So thank you very much for being here today, and it's a great pleasure to have George Sledge with me. You know, what I learned from the Precision Oncology Alliance meeting is I'm actually not going to try to introduce you because I noticed that didn't seem to be necessary. But I do want to confirm or disconfirm one thing. Because you're one of the rock stars of ASCO, is it true that Taylor Swift is going to have you do a set with her tonight?
You have obviously never heard me sing.
Okay, well, it's just circulating around, so I need to kind of understand that. So we're going to go through a few different areas, and I think we're going to talk about the Caris differentiation of the Caris approach as one part of what we're doing. We're actually going to start talking about how clinical genomics data in different ways can change how you might plan clinical trials, translational strategies, and kind of some other approaches. And what we'll try to do is we'll leave some time at the end for questions. And so if you do have some questions, we can kind of go through those probably in the last 10 minutes or so as we kind of go forward here. So maybe, George, just to begin, I know that Caris and ConcertAI have kind of come together, and we spent a lot of time thinking about our own partnership and relationship. And part of our enthusiasm was what was so differentiated about the approach that Caris was taking to molecular diagnostic testing. And I'm wondering if you wouldn't mind, maybe just as a little bit of a starting point, and you almost being slightly newer in your Chief Medical Officer executive role coming in here, how would you describe what's different in terms of the approach relative to other commercially available next-generation sequencing-based approaches?
Sure. Well, I guess if I may, let me start by why I came to Caris, because I think it's a good part of the answer to that question. I'm a lifelong academic. I spent 4 decades.
It went by quickly.
Yeah, it went by quickly. Yeah, in academia, and I throughout my career, I've been a data hog. Like many academicians, I loved having larger and larger data sets. And within the last decade, for the first time in the history of oncology, we're able to get data sets that are comprehensive, intense, data-rich, attached to clinical data in such a way that we can actually think about using it for the first time to affect the lives of patients. So for me, that's been one of the more exciting things in my life. I came to Caris in large part just simply because Caris, I thought, did it better than anyone else. If you look over the last decade, where for the first time we've been able to do next-generation sequencing, most of the work that has been done with limited panels that are in the tens or at most the hundreds in terms of the genes one is looking at. By and large, most of these panels have been DNA-based. Caris, several years ago, made the strategic decision, but I think also the patient-devoted decision that more data was better. And so that rather than working with a limited gene panel, we would look at the whole exome and the whole transcriptome. In other words, all 23,000 genes that make up every cell in our body, and the transcripts coming off of those genes. Having that data is incredibly powerful. No one knows where next year's drug target will be. So if you are stuck with a panel that is looking just at last year's drug targets, you're missing something. By definition, you're going to be missing something. Caris, in essence, have future-proofed things by virtue of just collecting so much data.
Yeah.
The ability to use that data, of course, has obvious research purposes.
Yeah.
Has obvious purposes in terms of being able to develop signatures in terms of therapeutics for which drugs will work in which patients. And these are all things that I think you can only do with large data sets.
Yes. So when you think about it, I mean, taking a full exome and transcriptome, and when you're thinking about it, this was actually only found in sort of smaller scale academic data sets, but without high depth in the clinical portion of the record.
Yeah.
What do you think what's the combined value as we start thinking about almost population scale, that depth on both for the genomic but also for the clinicals?
Sure, yeah, and again, I approach this from the past of having looked at decades worth of research where people would try and come up with a particular signature for, I'm a breast cancer doctor, how does hormonal therapy work in breast cancer? What became very obvious many decades ago is that if you look at a single gene, you never got the whole story, number one. Number two, if you looked at small populations, the likelihood that you would have a false positive when you did your data analysis was high.
Okay.
I mean, we for decades, we've lacked decent validation studies, so you have a literature polluted, and pollution is probably the right term for it, polluted with studies done by well-meaning people, but coming up with totally false results. Having large databases on the molecular side that are attached to large clinical databases allows you to get around many of those problems. There's an old saying that quantity has a quality all of its own. And I think that's absolutely the case when we're talking about molecular databases and their attached clinical databases.
So the word polluted and pollution is kind of a strong concept, and I can't kind of ignore that.
Yeah.
I kind of want to come back to that here for a moment. That would almost indicate that, without calling out obviously any one domain or study area, that some of what has been in the published literature that may constitute a body of kind of assumed insight
Yes.
may in fact need to be reconfirmed, or we may need to now take advantage of actually just a completely different scale and kind of depth of data and reconsider classes of questions and areas just because of that.
Indeed. Now, some of the failures are in essence sort of statistical failures. You know, the p-value was 0.05 on a population of 100 people, and the next study with another 100 people, it was 0.02. In the following study, it was 0.23, and so you never really knew.
Yeah.
Part of that is just simply a mathematical problem, but I think the other part is sort of a conceptual problem around biology, and that's to say, again, most of the history of our field is looking at single factors, at single genes, single mutations, single transcripts, and this fails to capture the complexity of biology, and in particular fails to collect the complexity of tumor biology, where we're having to deal with dynamic evolution of the tumor.
If I kind of go over to the nature of translational kind of sciences, we were talking a little bit about this before even we kind of start up the session. And if you're talking about a company that may be kind of taking a novel target, going into an area that may be implicated in multiple tumor types, how would you think about kind of beginning to use some of these data to both plan what those first-in-human kind of studies may look like, and how might you even think about changing the approach that you actually take to the clinical studies themselves? Because it sounds like the data we can now acquire, even standard of care tools, are actually providing more depth than even things that were being used in early phase clinical trials.
Sure. So I guess part of the answer to this is if you look at targeted therapies, therapies that form the basis of modern precision oncology, pretty much every drug that falls in this category has what I would call some form of gatekeeper biomarker. That is to say that you measure in breast cancer you measure HER2, or in lung cancer you're looking at a ROS or an ALK mutation. These gatekeeper mutations are what gets you in through the gate with your drug. But it absolutely positively does not tell you whether or not you're going to respond to that therapy.
Right.
Okay, so if you look at something like Herceptin for HER2 positive breast cancer, if you've got amplification of HER2 and you get Herceptin as frontline therapy, your overall response rate is 30%. Okay, it's gotten you in the gate, but most drugs fail most patients most of the time. This is the dirty little secret of medical oncology. Most drugs fail most patients most of the time. So that gatekeeper mutation identifies, more than anything else, a population that won't benefit from therapy, but doesn't tell you for sure who is going to benefit from therapy. And so collecting the hope, my hope certainly, and I think the hope of all of us in this field, is that collecting more data is going to get you closer to truth. It's going to get you closer to knowing who actually will derive benefit from therapeutics.
Yeah.
Listen, everyone in this room wants the best for patients. And
Sure.
the best for patients is a drug that works reliably, regularly in patients with minimal toxicity,
Right.
and not treating patients who won't benefit. And having more data, having more molecular data attached to more clinical data certainly is the best way to do that.
So, George, as I'm listening to you, and I'm kind of thinking about both, in the gate, but also understanding you're implying that there's a lot more that needs to be understood that actually will increase above that 30% that may have been kind of beneficiaries.
Yeah.
You could say that actually the process of that kind of in almost the sort of the translational spirit of really understanding that biological system should continue not just through the successive clinical development phases, but actually over the life of the drug now being available for patients themselves. How do you see that changing now with some of the tools that Caris is providing?
Well, so, I mean, just to give some simple examples, because these are real life examples. If we look at EGFR and lung cancer, okay, where we know that the first couple generations of EGFR drugs were inevitably thwarted by very specific mutations. Then you have a wonderful drug like osimertinib that comes along and takes care of that problem. Well, what you're actually seeing there is the evolution of the cancer, the mutational evolution of the cancer. More recent example in breast cancer, elacestrant. If you look at a newly diagnosed breast cancer patient with ER-positive breast cancer who's going to go on an aromatase inhibitor, and you measure for mutations in the estrogen receptor gene, ESR1, they are very, very rare. They're 1% or less of patients. You now put a patient on an aromatase inhibitor for 4 or 5 years, and then you look at them when they relapse, that number goes up to maybe 30 or 40% rate of ESR1 mutations, okay? What do you do now? You get an FDA approval for a drug that works for ESR1 mutations. That's elacestrant. Got approved about 3 or 4 months ago. I think we're going to see that more and more. And I think if you typically in terms of the life cycle of developing drugs, we've thought of in essence, in terms of patent life. But we also probably should be thinking, because the pharma companies I've spoken to over the years tend to have lots of wonderful scientists who are consistently working on a particular disease problem. Part of the life cycle development of a new drug probably should be, what's going to be the resistance to that drug and how can I thwart it, right? And to do that probably fairly early in the process, you need to be thinking about doing real-time molecular measurements of the ongoing evolution of the cancer. Now, in the past, of course, we've been kind of focused for a good part of what we've been doing in the molecular diagnostics revolution. We've done it off of solid tissue. Increasingly, of course, we're going to be able to do it off of liquid biopsies, which will probably be a whole lot easier for patients and a whole lot easier in terms of measuring the dynamic evolution of the cancer.
Yeah, I want to come back and I want to spend a little bit of time on the whole area of liquid tumor biopsies. But you said something that I think could actually have some reasonably profound implications for how people even think about what their strategies are and how I'm thinking about biopharma, because I think what you were kind of calling the gateway mutations probably takes a disproportionate share of the focus and emphasis of kind of most organizations. That's where a lot of the primary research came through. But what you're laying out here is if I take a longitudinal view and if I take an exposure-based view, I'm going to actually see a range of different transformations and new mutations of interest that may in fact be almost as significant as some of the gateway mutations you just identified.
Indeed.
And laying that framework out is not always how people start framing out kind of what they can do and the different strategies they can take and even necessarily what their pipelines look like. So that actually in itself I think is in a way I'd love to even follow up with you on that, because I think there's some ways that we could even kind of begin to publish out some views and information on that kind of material.
Yeah, indeed. And by the way, this isn't a criticism of the companies. This is
and that was a real comment, by the way.
Yeah, this is kind of more a reality of what's happened. And that's to say throughout most of the history of drug development, we just simply couldn't measure this stuff. We could not measure dynamic evolution of human cancers. We lacked the tools. They were too expensive. There were too few places that did it. there was no way to do it in real time in patients. This is a change that's occurred, in particular over the last, I would say, 5 years or so. And it actually allows us, I think, for the first time to think in terms of the overall lifespan of a tumor and of the patient who has that tumor.
You oversee, and I knew about the area of Caris's work in signatures, and I'll kind of let you maybe explain what that term actually means a little bit. I was not aware until actually I kind of saw your presentation actually that you have a portfolio that now is kind of under kind of your leadership now at Caris, but maybe you could give a little definition and also how the data and the information that you now have combined with almost new methodological quantifications and quantitative approaches are now enabling different kinds of decision support, et cetera. So maybe we could develop this a little bit.
Sure. Well, let's start with signatures. And this is sort of I won't say it's the opposite of, but the complement to the gatekeeper mutation approach. And that is to say that because tumor biology is typically complex rather than simple. Now, by the way, there are some drugs where it's simple. If you have a BCR-ABL mutation, the biology is pretty simple. The resistance mutations are actually in the same ATP pocket that the original drug tackles. Okay, so there are cases in cancer where the biology is pretty simple, where in essence they're the stupid cancers. Okay, but the problem is most cancers are smart, most cancers are complex, most cancers the genomics are complex. There's usually not a single reason for a cancer to become resistant to a drug. There tends to be multiple reasons. And because of that, you need to be thinking in terms of signatures, and that's to say not a single gene, not just a gatekeeper gene, but rather a whole bunch of different genes. How do you discover those? There's been a number of different processes that have used. So again, I'm a breast cancer doctor. Probably the most commonly used signature on the planet today is the Oncotype DX signature, 21 gene signature, developed with cutting-edge 2002 technology, where what then Genomic Health, now Exact Sciences, did was to look at basically the entire breast cancer literature and to pull out of that literature everything that had been looked at as a biomarker for breast cancer. And so they started with 300 different biomarkers. By the way, this gets back to the false positive problem in biomarkers. And then whittled this down to 16 cancer-related genes and then weighted them, put them together. That particular signature has resulted in roughly an 80% reduction in the use of adjuvant chemotherapy in breast cancer over the last 2 decades. Think about that. Think about the expense saved, about the toxicity saved. Women who don't have to throw up and lose their hair because of a molecular diagnostic. Okay, and in particular because of a relatively more complex signature. Now, the difference between then and now is that then, because you couldn't look at 23,000 genes simultaneously, you had to start with the list that came from the literature. You don't have to do that anymore. Now you get some tumor, either solid or liquid. You measure all of those mutations. You then have very smart mathematicians. At Caris, we have a wonderful artificial intelligence group that spends an enormous amount of time putting together complex signatures. We're presenting one of those at this meeting, if I could share that with you.
Yeah, please.
So this is one looking at who gets a brain metastasis. Now, for a very long time, one of the tragedies of cancer patients is that we'll do a great job of treating most of the body, but most of our drugs don't get into the central nervous system, and therefore patients will develop a brain metastasis and indeed go on to die of that brain metastasis. By and large, we don't know who those patients are. By and large, we've had no particular good way of monitoring them. We can't scan people every 3 months in their brain because it would be cost prohibitive and would drive the patients and the doctors nuts after a while. But what we think, based on our early studies with work done by Jim Abraham in our group, is that we now have the the molecular tools, and this, by the way, was developed using 220,000 patients, just to give you some idea, that allows us to very finely slice the population of solid tumors, not a particular tumor, but actually this is multi-cancer. And so we can identify populations that are at exceptionally low risk for ever developing a brain metastasis, and populations that are at quite high risk, maybe 3/4 of the patients will develop a brain metastasis in a year and a half. Now, when you think about, how might I use this? Okay, well, in the clinic, if I have a really high-risk population, maybe that's a population I screen more often. But if I'm in pharma and I am developing drugs that have CNS penetration, well, maybe I want something that will allow me to predict ahead of time who's at really high risk for getting a brain metastasis in the next couple of years. And then I can now use that population to test the drug's ability to prevent brain metastasis. And we, for the first time now, have drugs like tucatinib, like TDM1, that we actually know get into the brain and induce remissions in the brain. So wouldn't it be wonderful if we can't be thinking now in terms of specifically developing CNS penetrant drugs and then applying them to high-risk populations and seeing whether or not we might be able to benefit them? And I think, again, we think that, the signature we've developed might be a first step in that direction. By the way, this is a poster that Jim Abraham has here at ASCO this year. So we're very excited about that. But we're in the process of developing a bunch of different signatures. Yeah. Some of them are specific to particular drugs. Some of them are specific to particular conditions, like IO therapy across cancers, for instance. And we think just simply by having very, very large numbers of patients who've been treated and where we have appropriate clinical follow-up data, we should be able to come up with something exciting for many of these scenarios.
Yeah, there are definitely some companies that have even the logos I saw in this building that would be interested in that particular topic. But let me ask you, your comments about signatures, it's almost got its own parallel, because it sounds like that even if I have if I have an existing therapeutic entity, the ability to think through and use signature-like methodologies could add incremental value to different exposed subgroups and subpopulations and kind of aid in the management. Do you see the notion of signatures almost becoming an integral part of the life cycle of certain kind of oncology therapeutics, or how do you see that maybe evolving a little bit?
Yeah, I certainly hope so. Again, in the past, going back to the gatekeeper mutation analogy, because we didn't have follow-up data, we couldn't do this. Because we lacked the molecular data, because we lacked the systems to do this. What Caris does, our business model, by and large, is that docs send us tissues from patients who have advanced disease. Last year we got 100,000 samples in. We're on track, I think, to do around 140,000 this year. Having that sort of bulk data across large numbers of different cancers gives you lots more options to look at the evolution of cancers. I would add that it also gives you more options to see whether or not your drug works in cancers other than what it was first approved for. Because what frequently occurs out in the community is that a patient has a particular mutation. It's not one for which the drug was originally approved for, but the doctor says, what have I got to lose? Let's try this therapy. And then we're actually able to say, based on follow-up clinical data, is there evidence of benefit? And we've actually been looking at that in terms of, can you, in essence, create tissue-agnostic indications by using clinical data, from broad populations.
Yeah, the FDA actually has in some of the more recent guidance documents indicated that models that could become the basis of selection of a cohort may in fact be allowed for certain clinical trial designs. And you can imagine these methodologies actually may be a much more robust way, particularly if you're actually doing it, again, to your point, at population scale in terms of where that sits.
I mean, to give an example, because this is just sort of a thought experiment that we did, there's FDA tissue agnostic indication for pembrolizumab in IO therapy for tumor mutation burden high patients or DNA mismatch repair. We've looked at this, in essence, the same indication virtually for nivolumab, and have compared the time on treatment curves for pembro versus nivolumab in the setting of TMB high or DNA mismatch repair, and frankly, the curves are totally overlapping. You could think about, if you have a second or third drug in a class and the first drug has a tissue agnostic indication, could you, using real world scenarios, actually per the FDA guidance, think about using this sort of data to get a broader indication for the drug and therefore help more patients?
That should have some people's attention. So let me take you back to, I said we're going to kind of defer kind of the whole liquid tumor biopsy, and I know maybe here too, Caris is taking a differentiator approach, but maybe a short description on kind of why a differentiator approach here, but also as you overlay now kind of this ability to have liquid tumor data and maybe both in adjuvant, neoadjuvant, and kind of later phases, what are you seeing the implications here of at least early views? So,
sure.
I'm going to carefully frame it that way.
Yeah, so liquid biopsies, and if we define liquid biopsies, in essence, is using next generation sequencing of either DNA or RNA. Liquid biopsies have the potential to be used across the entire spectrum of cancer. They have the potential to be used in an early diagnostic setting, all the way to late management of patients with metastatic disease. And at every step along the way, you can imagine the use of liquid biopsies. Patients don't like having needles stuck in their liver and lungs repetitively. Patients, humans, don't like having needles stuck in their lungs and liver repetitively, and doctors don't like doing it because every time you do it, there's a potential for hazard. That hazard doesn't exist by and large sticking a needle into someone's vein. So the potential to monitor is much easier at a patient level, and therefore potentially much more utilitarian in terms of doing repetitive measurements to look at tumor evolution over time. And so I think that's the promise of liquid biopsies. Liquid biopsies have their own issues. We also will be looking at the buffy coat, the white blood cells, and off of the buffy coat, we'll be looking at the host genome, and we'll also be looking at what's called chip subtraction. Clonal hematopoiesis, that's to say mutations that are basically occurring in your bone marrow that are pumping out white blood cells and which increase with age, are really common. We already know, based on published data, not our data, but other folks' data, that, for instance, if you look at a wonderful paper in Clinical Cancer Research a couple of years ago on this, if you look at prostate cancer, which is old men. Okay. If you look at prostate cancer, a significant proportion of older men have clonal hematopoiesis that actually are mutations that would lead you to give a patient a PARP inhibitor for prostate cancer. In other words, treating a patient's circulating white blood cells for something that you think might be going in their metastatic prostate cancer tumor, and it's not. So clonal hematopoiesis, if you do a clonal hematopoiesis subtraction, which we're doing as part of this assay, this allows you to avoid treating patients who will not benefit from the drug. So we think that's going to be a significant addition. So this is currently at MolDX awaiting regulatory approval. So we hope that we'll be rolling it out in the not too distant future. But this sort of approach, hugely increasing the amount of data you can get if you're trying to track the evolution of a human cancer, I think will be great. But also from a safety standpoint, doing the subtraction of clonal hematopoiesis will increase the safety for patients with cancer.
Do you see actually, as you're talking and also thinking about this longitudinal view and also understanding what response looks like and patterns of resistance look like, do you see integrating this early on in a variety of new drug development programs?
Sure. Yeah. I mean, starting at the very beginning.
Yeah.
So newly diagnosed patients.
Yeah.
One of the great white whales in all of oncology has been cancer prevention.
Yeah.
It's really, really hard to do cancer prevention trials. I mean, they're expensive, they're time-consuming.
Yeah.
A large amount of the expense and time-consuming is that if you start with the general population, not many of them actually get cancer.
Good.
So you have to treat a lot of people to benefit a few patients. If you could come up with molecular diagnostics that would allow you to identify those patients regularly, well, then we'd be able to tighten up the population and make those trials cheaper and quicker. At the time of diagnosis, a woman comes in and has a mammogram, someone comes in, has colonoscopy. If on the day of diagnosis, you could already tell what mutations are present in their cancer, well, maybe now you're talking about thinking in terms of molecularly based neoadjuvant therapy starting on the day of diagnosis.
Right.
I mean, the opportunities here, I think, are incredible.
So actually what I'm hearing in that one is historically standard of care tools and standard of care practices probably wouldn't really support operating that way, but actually as this now gets more integrated into it, and if we actually have any reason to suspect any form of risk, and we could use a variety of other tools, we've talked about digital H&Es and other things, there may be ways of using current standard of care to then accelerate where that testing may bring benefit and where those interventions
Indeed.
But we have some work to do on that.
This is early days for many of these things. So whenever a former academic promises you something, you should ask for several reality checks. But this is,
Was that a promise?
The excitement is palpable.
Yeah, that's for sure.
I mean, every session you go into has a new phase 3 trial for a novel agent. That's a game changer, right?
Yeah.
And a large amount of that excitement comes from the fact that this really is approaching real precision medicine.
Yeah.
That we are at a patient level.
Yeah.
Actually going to be able to say, Mrs. Jones, you have lung cancer type 3A, and type 3A benefits from this drug, and plan B is that once we notice the evolution of your cancer to have the mutation, boy, we got another drug for you.
Yeah.
That has been the hope and promise in this field for ever so long, and I think the combination of molecular diagnostics with novel therapeutics actually is getting us close to that.
I know that one of the reasons why our two organizations kind of came together was to start creating data sets that of a scale and scope that actually further support that, and even being able to get down to narrower populations, but with statistically significant numbers of patients still in them.
Indeed.
And with a depth that actually kind of allows us now to be actionable, and whether it's taken into signatures or other work.
Yeah.
That's super.
We want quantity of data, we also want quality of data.
Absolutely.
And so, one of the things that we're particularly excited about working about ConcertAI with is that it significantly improves the quality of the data that we work with from a clinical standpoint. Those sorts of combinations, high-level molecular data, high-level clinical data, get you a lot closer to where you need to be.
Right. So I do want to offer, if anybody has any questions, we're happy to take some. Yeah.
George. Just connecting a few of the topics you cover, and you're a breast cancer physician. Just maybe to get a bit more specific, like drug conjugate, ADCs, right? It's a big topic.
Yeah.
And you take an HER2 and this now antibodies bring you to the right cells, sparing healthy tissues, so they're very effective. So on one side it looks like this is a major advancement, but on the other hand, you may actually create a veil to pierce through to, because then you need to really understand resistance mechanisms. What would be your advice to those who are already treating patients with these drugs and those that are developing?
So I've thought about this a lot. In breast cancer, we have drugs like trastuzumab deruxtecan. We have sacituzumab govitecan, unpronounceable drugs, right? These they're fascinating in part because they have specific molecular targets, TROP2 or HER2. So you're using the antibody for that. You're attaching, in essence, a cellular poison to that. What's the drug resistance based on? We have 2 decades of research in HER2 about mechanisms of resistance to HER2-targeted therapies. Are those mechanisms still relevant in an era of antibody-drug conjugates, or is the resistance now basically chemotherapy resistance to the payload? And we don't know is the honest answer. So how are we going to learn that? I think, again, I would think that having large numbers of patients who've been treated with one of these agents and then looking at what happens to the tumor may be the best way of looking at that and allowing us to tease out, is if I give sacituzumab govitecan, does resistance occur because the tumor is no longer expressing TROP2? Or does resistance occur because it's TCAN resistance? And we know a lot about TCAN resistance. And I think the only way, outside of cell lines in a laboratory, which have their own limitations, the only way that we're going to learn about this is to look at fairly large populations. So I think it's a great question, and it's one I've thought a lot about.
We may have a follow-up conversation for you on that topic.
Indeed,
But,
indeed.
I know there was one.
Tumor evolution, you mentioned it a lot. How long is a sample relevant to the patient? Because the diagnostic molecular profile is not going to be the same after two rounds of heavy-duty DNA shredding therapies. So I know we were talking liquid biopsies, but putting that aside, is there a way to avoid needing a relevant sample from a biopsy?
Yeah, it's a really great question. So I don't know that I can give you a final answer, but I can give you an initial answer, and that is to say that if you look at the correlation between a tissue and a liquid biopsy, and because we've done this as part of the development of Caris Assure, what you find is that there's a very high correlation between what you see in terms of, at a mutational level in the first 90 days or so. Okay, so if you have the solid tissue biopsy and the liquid biopsy within 3 months or so, there's a really tight correlation between the mutations you see. Boy, but you get more and more drift the further and further out you get. And again, this is exactly what you're saying. That there's intervening therapies. Those intervening therapies are affecting the makeup of the tumor. They're probably reducing sensitive clones. They're leaving resistant clones, and so the mutational makeup changes pretty quickly. What this tells us is that if we're serious about this, we probably need to start measuring reasonably early, and may ultimately need to measure reasonably often. I want to be careful about saying this. That this is not George Sledge's new standard of care that you need to get Caris Assure every 3 months for the rest of your life, because I think this is going to be very tumor by tumor, and we're going to actually need to accumulate the data to demonstrate that this is actually beneficial to patients. One of my mantras is that finding bad news early doesn't turn it into good news. Okay, it's only useful to do something to find the bad news if you can actually alter a therapy and benefit the patients. And again, this is going to be very tumor by tumor, very case by case.
Well, even just to further support at least the merit of the debate you were having is, even some of the ASCO leadership have been kind of commenting and kind of beginning to muse that may actually become more of a requirement to really change outcomes.
Yeah,
so
and the ASCO guidelines are clearly starting to move in that direction.
So.
Yeah.
So I want to thank everybody for taking the time to be here. I'm super glad that we've got this recorded. I really appreciate your perspectives, and we really appreciate where the partnership can go too. And I know you've already identified some research projects I feel like I owe you some follow-up on, so we will kind of get that kind of going underway as well, with pleasure, actually.
Yeah.
Okay.
Thanks
so
for
thanks
having
very
me.
much. Thank you.
Thank you.